Today I'll be talking about our poster entitled In Vitro Micronucleus and Comet Chip with Metabolically Competent HepaRG Cells. The in vitro micronucleus assay is a well-established cytogenetic assay for mammalian cells and is part of the regulatory battery recommended by the ICH and the US EPA. The Comet assay is a tissue-based DNA damage assay that has an in vivo guideline by the OECD that has been recently established. So these are two well-known genotoxicity assays that we have employed in HepaRG cells. In collaboration with MIT, we have developed a medium throughput inversion of the Comet assay that uses Comet Chip technology. This Comet Chip technology was developed at MIT in the laboratory of Bevan Ingleward, and we have adopted this technology to the metabolically competent HepaRG cells. The traditional mammalian cell genotoxicity assays use highly induced rat liver S9 that is supplemented with cytochrome P450 cofactors. Phase 1 metabolism, but there's not any phase 2 metabolism in this S9. Therefore, we feel that using HepaRG cells, we can have both phase 1 and phase 2 metabolism. Phase 2 metabolism is very important for the detoxification process. Differentiated HepaRGs at a low density are both hepatocyte and biliary cells that create a natural co-culture system. These differentiated cells do have metabolism capabilities, for instance, phase 1 and phase 2 metabolism, nuclear receptors, and drug transporters similar to cryopreserved human hepatocytes. They've also gone recently an ECVAM validation trial showing that HepaRG cells are as reliable as an enzyme induction model as cryopreserved human hepatocytes. The two genetic toxicology endpoints we are using in HepaRG cells are the micronucleus and DNA damage using the Comet assay. The chromosomal damage we are measuring in HepaRG cells is done using the in vitro microflow micronucleus assay developed by Latron. This assay detects relative survival, apoptosis necrosis, cell cycle, and a measurement of the micronucleus frequency. This assay also requires cells to divide once, so we use epidermal growth factor at the end of exposure to stimulate these cells to undergo one division for the micronucleus assay. The Comet Chip assay, as developed at MIT, uses a bed of micromeals within the agarose gels in a 96-well plate. Therefore, each well of a 96-well plate contains more than 200 microwells. Then cells are deposited into these wells, and they settle into these microwells within each well, allowing the in situ Comet assay to be conducted within those chips. This Comet chip has many advantages over traditional Comet assay. It's time saving. For example, the traditional Comet assay for one single tissue takes 4 days of technician time. With the Comet Chip assay, you can analyze 96 samples in 2 days. It's very fast and it's automated. It can be automated to perform a number of assays in 1 week that you could not do in the traditional Comet assay. The experimental design we use uses a 3-day exposure regimen. Cells are exposed for 3 days, 24 hours apart. Then at the end of the 3-day exposure, we either add epidermal growth factor for the micronucleus assay. We also measure ATP levels as a measure of cytotoxicity, and then we take each of those wells and transfer those wells to the Comet Chip, where we're able to do the Comet assay using this high throughput Comet Chip technology. The data thus far on more than 60 compounds has demonstrated that the micronucleus assay with the compounds we have tested there shows about 100% specificity and 100% sensitivity. So it detected the genotoxic compounds that we tested. In this case, it was 8 genotoxic compounds and 6 non-genotoxic compounds, and they responded appropriately in the micronucleus assay. In the Comet Chip assay, we showed with a small set of prototype compounds that the response is very similar to what we get from the traditional Comet assay using Comet Chip and Comet standard slides, 2-well Trevigen slides. Therefore, we went forward and analyzed more than 60 compounds in the Comet Chip assay belonging to a number of broad categories including genotoxic compounds, non-genotoxic compounds, compounds that are positive in the Ames or negative in the Ames. We showed that they have a very good response. The responses are very consistent with the expected outcomes. In addition, we were able to demonstrate that some of these compounds induce cytochrome P450s as part of their genotoxicity profile. For example, cyclophosphamide was shown to cause a dose-dependent increase in 3 P450s, including CYP2B6, 3A4, and also induced a glucuronidtransferase 1A1. These assays incorporating HepaRG cells can be potential follow-up to results from S9-based assays and regulatory assay testing to reduce reliance on animal studies. This funding was supported by the Small Business Innovative Research Grant from the NIEHS.